Thank you. So before I begin, I'd like to also thank the meeting organizers and certainly ARM for the wonderful opportunity to share a little bit about our company. So far it's been a wonderful conference, and hopefully everyone's enjoying the interaction. As mentioned, my name is Lisa Deschamps, and I'm the CEO of a private company called AviadoBio. Our company is London-based, globally focused, and we are a CNS gene therapy company. Prior to joining Aviado, I spent over 25 years at Novartis, where my last role was leading the global neuroscience business, had the wonderful privilege to do the acquisition of Avexis, and then lead the worldwide commercialization of Zolgensma, up until 41 market approvals, 29 reimbursements, and most importantly, 2,000 patients. So that is certainly what inspired me to come here and work, just like all of us are, bringing more of these transformative therapies to the patients who definitely need them. A little bit about Aviado. So Aviado, as mentioned, is a CNS gene therapy company. Our mission is to develop and deliver transformative gene therapies to patients in need. We are starting in neurodegeneration. We have a program in frontotemporal dementia, and then our pipeline consists of being focused today on ALS. I'll tell you more about that in a second. The impetus, or again, the inspiration behind the start of the company, Professor Chris Shaw, for those of you who may know him, he's from King's College London. He's a neurologist, neuroscientist. He's been treating dementia and ALS for over 30 years. And in Chris's words, he identified genes, led over 70 clinical trials, treated patients in the clinic hands-on every day, and has made a lot of empty promises to generations of families. And so before he retires, he wanted to be part of the solution. So Chris and two other scientific founders kind of hit the ground looking for funding just a couple of years ago. The company was only started in September of 2019, and it is a spin-out of Chris's lab at King's College London. Again, our mission is to bring gene therapies across CNS disease, and our focus is really on delivery. So I know many people are focused on delivery today, especially as it relates to neurological conditions, and I'll tell you a little bit about our approach to that. But we really believe in order to have, to maximize the therapeutic benefit for patients and do this in a way that is safe and tolerable, we need to be both a drug and a delivery company. To date, we've raised a little shy of $100 million. So our seed round was $16 million. And then at the end of 2021, time goes fast, we raised a Series A of $80 million. So to date, we're a little shy of 100. Our platforms are being optimized for both gene supplementation as well as gene silencing. And our lead program is in frontotemporal dementia. Some people don't know what that is, although since Bruce Willis's diagnosis recently, a lot of people have asked me more about frontotemporal dementia. But it is one of the most common forms of dementia in patients under the age of 60. So this disease primarily affects people very young, and it is the second form of dementia to Alzheimer's in overall. So again, it's a quite common form of dementia, but still a lot to be done as far as education and awareness overall. We received last year orphan designation in both the EU and US, and our lead program, again, is in frontotemporal dementia, but in patients who have a specific subtype. So they have a mutation in their GRN gene, so it's a monogenic disease. We have currently spent the last couple of years building the organization, obviously to be able to successfully advance not only the lead program, but also our pipeline. We have 56 people currently. Again, we are London-based, and we're co-located as well in the US, but for sure globally focused. For many of you, if you don't know, but I'm sure most of us in this room do know, given the scientific backgrounds, but biodistribution is a key challenge in treating CNS diseases. At a very young age, we develop our blood-brain barriers, which are meant to protect ourselves from any foreign substance. So allow nutrients in, but really not allow foreign substances in, which makes delivering drug to the brain and the spinal cord very difficult. And so what we've seen, certainly in gene therapies across CNS disease, is a lot of failure today, some successes, certainly like Zolgensma and others, but a lot of failure because trying to deliver doses that can penetrate the blood-brain barrier and get into where they need to go at levels that are not toxic has been a key challenge. And so our focus is really solving this challenge and looking at much more direct delivery approaches to be able to enable and do this effectively. So we define our focus on delivery in kind of three key ways. One is around the constructs themselves and making sure we optimize the constructs to be targeted, efficient, neuronal specific. Second area is around the delivery itself. And here we look at routes of administration. I'll tell you a little bit more about that in a second, as well as the delivery technologies that are used to enable the very targeted routes of administration. And then lastly, but not leastly, certainly the expertise and the capabilities that it takes to be able to do this successfully. So we have a team of neurosurgeons that are employed with the organization, and that coupled with the neurologists and working with the community is where we spend most of our time. Our pipeline is focused, again, on both supplementation and silencing, knockdown. And this is what it looks like as it relates to both the delivery approach as well as the particular therapeutic areas. So our lead program is the frontotemporal dementia. As you can see, we're currently in just coming out of preclinical, so we've submitted to the regulators, and we're waiting to start our clinical trials, hopefully as early as August, September of this year. I'll tell you a little bit about that at the end. We're also looking at, and that has an intrathalamic delivery approach, which means we're going direct intraparenchymal, so direct into the brain, but specifically into the thalamus, because we believe that the thalamus is a central relay hub to the brain, and many of these diseases, while they start kind of locally, they're global in nature, and you need to be able to distribute them evenly across the regions of the brain. In our pipeline programs, we are focused in ALS, and we have 4 targets, as you can see there on the screen. Progranulin, TDP-43, SOD1, Ataxin-2, as well as C9. And they're all at different stages. Most of them are in early discovery. We have some key data readouts in vivo this summer. So very soon we'll know where we're headed as it relates to the pipeline as well. For the pipeline assets, we're looking at also a direct delivery approach, which is called sub-peel. So this is going in direct into the spinal cord, lifting the dura, and providing, again, a very direct delivery approach into the spinal cord. We've announced a couple of weeks ago an exclusive partnership with a company called Neurogain. You may know Martin Masala from UCSD out in San Diego, and Martin and his team have developed this sub-peel delivery approach along with a proprietary needle that enables the ease of the delivery and the surgery. So we have exclusive rights in CNS disease to use this delivery approach not only for our own programs, but also to sub-license for other companies in their areas of interest that are, of course, non-competitive to ours. We chose FTD as the lead candidate for a couple of reasons. One, the significant unmet need that was just mentioned. So again, almost 100,000 people across the US and Europe in particular, but it goes well beyond this, of course, have frontotemporal dementia. About 10% of that has the specific mutation, so the monogenic mutation of GRN. And we think also progranulin plays a much broader role across many neurological conditions, certainly neurodegenerative conditions, but more to come on that. We also know that it's got a very quick-moving decline. So unlike Alzheimer's, which can take many years to develop and see symptoms, this disease, once the symptoms start, moves very rapidly. So from a clinical trial perspective, that enables, obviously, it's sad for the patients, but enables you to see how your product is working and be able to take endpoints and measurements and biomarkers that are established in this space. Some stats show that patients from diagnosis die as quick as 5 to 7 years. The reference we use here is 6 to 12. There's a lot more to really be known about this disease. Although we do have natural history, we still have a lot to know longitudinally as we continue on. And then there's obviously, given the age of the population that it does affect, there's a significant cost burden and healthcare utilization burden overall and societal impact associated with, people who are in their 30s and 40s who should be working and having a very much optimal quality of life at that stage. So it really, again, affects patients in much more prime of our lives. From a disease biology perspective, again, with the subtype we're going after, it's got a very clear-cut biology. We know the pathogenesis. We know it's monogenic. We know we need to affect and restore progranulin levels. So for a normal, healthy person who doesn't have this disease, the people who do have this disease have about 20 to 30% of progranulin versus us who have, whatever normal is for us. So we know restoring progranulin is the key to bring patients back, or certainly to prevent further progression from happening. We know that progranulin decline also increases and affects the accumulation of TDP-43, which again is not only in FTD, but really affects more broadly in neurodegenerative diseases. So again, by restoring progranulin levels, you will also have an effect on the accumulation of TDP-43. From a tractability perspective, there is natural history data, both in the US and Europe and some parts of the rest of the world. So again, we're able to at least have a baseline of what, good could look like and what the normal trajectory is of this disease so far. It is measurable by MRI as it relates to atrophy and other effects that we see in the brain, and we know we have some already established biomarkers in the space. There are currently no approved therapies in FTD, although there are a lot of people trying. So there's a number of gene therapies as well as non-gene therapies that are currently in development across the board, different phases. As mentioned a couple minutes ago, with our lead program, we're going intraparenchymal, but specifically through the thalami. We believe that the thalamus is a key hub for biodistribution in the brain, and by exploiting this, we can exploit the neuronal networks needed, again, not only to address the disease locally, but also to address the disease globally as it progresses in severity. We have to date data across multiple species, so our non-clinical and then early and then preclinical work includes data in mice, in sheep, as well as in non-human primates, and all of the data has been extremely consistent, showing really nice broad biodistribution, which we believe ultimately will translate into a maximal therapeutic effect without any of the baggage that we've seen across other programs from a safety perspective. So we recently presented data last year at ASGCT and ESGCT, where we showed data from, again, at least the mice and the sheep. The NHPs hasn't yet been published, where we showed that we deliver a superior biodistribution without anything in the liver or the serum from a safety perspective. I didn't put the data in the presentation today just for the time, but certainly if any of you want to see it, I'm happy to send you the abstract. This is a little bit about our clinical trial. So obviously all the work that we did from non-clinical as well as the regulatory enabling studies in primates have enabled us to be able to submit to the regulators and to be thinking about starting our clinical trial as early as this summer. The trial design is an open-label, dose-escalating phase 1/2 study. Primary endpoint, of course, is safety. Will be a safety mark at 6 months, and the study will continue on for 4.5-year follow-up. The rest of the endpoints, again, will include other measures where we'll be able to also look at not only biomarkers, but also efficacy measures around cognitive decline, et cetera. What we don't know today is how long it will take to see those other measurements. We know that, the surgery itself will see early acute procedural type assessments, right? Was it safe? How did people respond through the surgery, et cetera? But obviously over time, we'll be looking for obviously efficacy type measurements, functional type of benefit, which probably will take anywhere between 6, maybe more likely 12 months to start to see those other benefits over time. We'll be starting the trial in in Europe, and then moving into the US subsequently. It's meant to be one global study with multiple cohorts, obviously looking at the parameters as defined and defining dose escalations as we go forward. It is a stereotactic neurological procedure, so it is surgery. And so we're using the ClearPoint system. Many of you might know the ClearPoint system. It is registered in US, Europe, and other places. And we're doing this, obviously, it's an MRI-guided system, so you're able to see where you're going, which is important when you're going into the brain. And obviously, it's an established delivery technology that has been used across multiple other areas. We are also, as it relates to trial design and thinking about how we execute this effectively from an operational perspective, we have a hub and spoke model where we are going, when you think about, obviously, for a neurological surgical procedure, you need not only the treating neurologist, but you need a surgical site as well as referral neurologies sites kind of, feeding those patients in. So it's a bit of a hub and spoke type model. Again, we're starting in Europe first, and then we'll be moving subsequently into the US. So to date, we have EU and US, in summary, orphan designation received. We've so far submitted 8 regulatory packages across Europe and the UK, and we'll be submitting the IND by the end of this year. We've got both 3-month and 6-month tox data in non-human primates, as well as a biodistribution study as well in non-human primates. We've got all of our manufacturing completed and our GMP batch ready to go for the clinical trial. We've got compatibility studies done as it relates to the device. We've already presented, key preclinical data, as just mentioned before, and we are in the process of hopefully starting that trial as early as August, September, pending the regulator feedback, but so far so good. And we hope to then have some more meaningful clinical data as early as Q1 2024, assuming we start as planned. This is a little just overview of the team. Pretty much everyone on the team has worked in gene therapy in some way, which I feel very proud to be a part of, whether it was in the labs or academia or commercialization, CMC, tech ops, et cetera. So everyone, all 56 employees to date, have gene therapy experience across some part of the life cycle. Those were our investors as well on the right side. We have 8 investors currently, 7 VCs, and 1 private equity, which is EQT, the largest European private equity company. So in summary, we are set to kind of what we hope to be delivering the future of gene therapy in neurological diseases. We feel like we've got the right approach, focusing on our delivery aspects as just shared, to really be able to, again, maximize the therapeutic benefit for patients and at the same time minimize any safety negative effects that we might see. So we are on our way, and hopefully next year I'll have more to share. So thank you.